Chapter 8. Cell. The unit of life. Part 2. A prokaryotic cell. Cell envelope. Flagella. Pili. Fimbriae. Ribosomes. Inclusion bodies. Imagine a living cell. No nucleus. No mitochondria. No Golgi body. No endoplasmic reticulum. Yet, this tiny cell has ruled the earth for billions of years. It lives inside hot springs, oceans, soil, even inside our own body. It multiplies rapidly, adapts quickly, and is one of the simplest yet most successful forms of life ever created. How is that even possible? Today's video is all about understanding that incredible cell, the prokaryotic cell. We'll explore its protective covering, its movement, its protein factories, and even the tiny storage granules hidden inside it. And by the end of this video, you'll realize that simplicity doesn't mean weakness. So, let's begin. What is a prokaryotic cell? Before understanding its parts, let's first understand what exactly a prokaryotic cell is. A prokaryotic cell is one of the simplest types of living cells. Unlike the cells present in plants and animals, it does not have a true nucleus. It also lacks membrane-bound organelles. Even though it looks simple, it can perform every basic life process needed for survival. That's why prokaryotic organisms are among the oldest and most successful life forms on Earth. Now let's understand how such a tiny cell is organized. Cell envelope. Let's begin with the outer covering. Imagine you're traveling on a bike. First, you wear a raincoat. Inside the raincoat,
A jacket.
and inside that, your shirt. 3 protective layers, working together. Similarly, a prokaryotic cell is also protected by multiple coverings. Together, these coverings are called the cell envelope. According to our diagram, the cell envelope consists of 3 parts: plasma membrane, cell wall, and in some organisms, a capsule. Each layer has a different responsibility. Let's understand them one by one. Plasma membrane. The innermost layer of the cell envelope is the plasma membrane. Think of it as the security gate of the cell. Every person entering or leaving a building passes through the main gate. Similarly, every substance entering or leaving the cell passes through the plasma membrane. That's why the plasma membrane is called selectively permeable. It decides what should enter and what should leave. Besides transport, many important metabolic activities also take place here. So remember, the plasma membrane is much more than just a covering. It is an active working surface of the cell. Cell wall. Outside the plasma membrane lies the cell wall. Imagine trying to inflate a balloon without any outer support. It can easily burst. Similarly, water continuously enters bacterial cells through osmosis. Without a strong outer wall, the cell could burst. That's why the cell wall acts like a rigid protective shield. It performs 3 major functions. First, it gives the cell its shape. Second, it provides protection.
Third,
It prevents osmotic lysis, which means bursting due to excess water. According to our mind map, the cell wall is made of peptidoglycan. That single word is extremely important for exams. Whenever you hear bacterial cell wall, immediately remember peptidoglycan.
capsule.
Now let's move to the outermost covering. Notice something written in the diagram. It says, capsule, if present. That means, not every bacterium possesses a capsule. Some do, some don't. Whenever present, the capsule forms the outer sticky covering of the cell. Its major function is protection. It helps bacteria survive drying conditions. It also protects them from phagocytosis. In simple words, it makes it difficult for our body's defense cells to destroy bacteria. Because of this, the capsule also plays an important role in pathogenicity or disease-causing ability. So whenever you see a bacterium with a capsule, think of it as wearing an extra protective jacket. Quick revision. Let's revise the cell envelope before moving ahead.
Capsule.
Outermost sticky covering. Protection. Helps prevent phagocytosis. Important in pathogenicity. Cell wall. Made of peptidoglycan. gives shape, provides protection, prevents osmotic lysis, plasma membrane, selectively permeable, controls entry and exit, site of many metabolic activities.
Simple. 3 layers.
3 different jobs: Flagella, Pili and Fimbriae. Now here's an interesting question. If bacteria are so tiny, how do they move? Do they have legs? Do they have muscles? Of course not. Instead, nature has designed 3 special surface structures:
Flagella.
Pili and Fimbriae. Students often confuse these 3. Let's remove that confusion forever.
Flagella.
Look carefully at the bacterial cell. Notice the long whip-like tail extending outside. That is the flagellum. Its main function is movement. Whenever a bacterium wants to swim through water, the flagellum rotates and pushes the cell forward. Think of it like the propeller of a boat. No propeller, no movement. One more important point. Flagella are composed of a protein called flagellin. So remember, movement,
Flagella.
protein.
Flagellin. Pili.
Now look again. Pili are much longer than Fimbriae, but they are fewer in number. Unlike flagella, they are not mainly meant for movement. Instead, their most important role is conjugation. Conjugation simply means transfer of DNA from one bacterium to another. Imagine 2 friends exchanging important notes before an exam. Similarly, 2 bacterial cells can exchange genetic information through Pili. So remember one keyword. Pili is equal to DNA transfer. Fimbriae. Now comes the third structure.
Fimbriae.
These are completely different. They are short,
Thin.
hair-like, and present in very large numbers. Their job is not movement. Their job is attachment. They help bacteria stick firmly to surfaces. Whether it's a rock, a tooth, or body tissues, Fimbriae help bacteria remain attached. They also play an important role in biofilm formation. So remember,
Flagella. Movement.
Pili, DNA transfer, Fimbriae,
Attachment.
3 structures, 3 completely different functions.
Ribosomes.
Now let's move inside the prokaryotic cell. We've understood its protective covering. We've seen how it moves. Now here's a question. Where does the cell make its proteins? Because remember, every living cell needs proteins. Proteins help in growth, repair, enzyme formation, and almost every life process. So where is the protein factory? The answer is
Ribosomes.
think of ribosomes as tiny manufacturing machines working continuously inside the cell. They read the genetic instructions and assemble amino acids together to produce proteins. That's why ribosomes are known as the site of protein synthesis. Now look carefully at the diagram. You'll notice 3 important points. First, prokaryotic cells contain 70 S ribosomes. The letter S stands for Swedberg unit, which represents the rate at which ribosomes settle during centrifugation. For now, you only need to remember one thing. Prokaryotes have 70 S ribosomes. Second, these ribosomes are smaller than the 80 S ribosomes found in eukaryotic cells. We'll understand eukaryotic ribosomes in the next part. For now, just remember this simple comparison. Prokaryotes 70 S. Eukaryotes 80 S.
Third.
Unlike many organelles, these ribosomes are free in the cytoplasm. They are not enclosed by any membrane. Instead, they remain scattered throughout the cytoplasm, continuously producing proteins whenever the cell needs them. So if someone asks, which structure is the protein factory of a prokaryotic cell? Your answer should immediately be
Ribosomes.
memory trick. Here's a simple way to remember. Think of the number 70. Imagine a factory with a huge board outside saying, Factory number 70. Inside, workers are constantly making new products. Similarly, 70 S ribosomes are the protein factories of prokaryotic cells. Simple,
Visual.
and easy to remember. Inclusion bodies. Now let's move to another interesting structure. Imagine you're preparing for an exam. Would you keep all your food in your stomach forever? Of course not. You store food in your kitchen. And whenever you're hungry, you use it. Similarly, cells also need a storage system. That's where inclusion bodies come into the picture. Inclusion bodies are simply storage granules. Their job is to store reserve substances so that the cell can use them whenever required. Now look carefully at the diagram. One important point is written there. They are not bounded by a membrane. This means, unlike many organelles found in eukaryotic cells, inclusion bodies do not have their own surrounding membrane. They are simply stored directly inside the cytoplasm. Now let's look at the different types shown in the diagram. Glycogen granules. The first one is glycogen granules. These store carbohydrates. You can think of them as the cell's emergency energy reserve. Whenever energy is required, the stored glycogen can be used. Polyphosphate granules. Next, polyphosphate granules. As the name suggests, these store phosphate compounds. Phosphates are essential for many important cellular activities, so the cell stores them for future use. Lipid granules. The third type is lipid granules. These store fats. Lipids serve as another important energy reserve. Whenever required, the stored lipids can be utilized. Sulfur granules. Finally, sulfur granules. These store sulfur-containing substances. Certain bacteria require sulfur for their metabolism. So instead of searching for it every time, they simply store it for later use. So although these 4 granules store different materials, their basic purpose is exactly the same. Storage. That's why they are collectively called inclusion bodies. Easy memory trick. Whenever you hear inclusion bodies, think of a storeroom. A storeroom can contain food, tools,
Fuel.
or any useful material. Similarly, inclusion bodies store different reserve substances that help the cell survive whenever resources become limited. Quick recap. Now let's revise everything we've learned today in just a couple of minutes. A prokaryotic cell is a simple, unicellular cell that does not have a true nucleus or membrane-bound organelles. Its outer covering is called the cell envelope, which consists of the plasma membrane, cell wall, and in some bacteria, a capsule. The cell wall is made of peptidoglycan. It gives the cell its shape, protects it, and prevents osmotic lysis. The capsule, when present, is the outermost sticky covering. It protects the bacterium from drying and phagocytosis and increases its pathogenicity. The plasma membrane is selectively permeable. It regulates the entry and exit of substances and serves as the site of many metabolic activities. For movement, the cell uses flagella, which are long, whip-like structures made of flagellin. For DNA transfer during conjugation, bacteria use Pili. For attachment to surfaces and biofilm formation, they use Fimbriae. Inside the cytoplasm, the cell contains 70 S ribosomes, which are smaller than eukaryotic 80 S ribosomes, remain free in the cytoplasm, and act as the site of protein synthesis. Finally, the cell contains inclusion bodies, which are non-membrane-bound storage granules. Depending on the material stored, they may be glycogen granules, polyphosphate granules, lipid granules, or sulfur granules. Every one of these structures works together to help this tiny, simple cell survive, grow, and perform all the essential functions of life. Today, we explored one of the simplest yet most fascinating cells on Earth, the prokaryotic cell. Even without a true nucleus or membrane-bound organelles, it performs every essential life process with remarkable efficiency. But now it's time to move one step further. If prokaryotic cells are the simplest form of life, what about the cells that make up plants, animals, and even our own body? How are they different? What makes them more advanced? In the next part, we'll step inside the eukaryotic cell, explore the plasma membrane, understand the fluid mosaic model, learn how substances move across membranes through different types of transport, and uncover the fascinating process of endocytosis. So I'll see you in part 3. Until then, keep learning, keep revising, and I'll see you in the next one.